Stop it. Hey, Daddy, we're just gonna get
Well, how about an orange?
orange. How about kiwi?
Um,
Kiwi.
I don't really know how to.
It's kind of the same as an orange or an apple.
Just cut it.
Yeah, just cut it in half. Just cut.
Cut it in half, and then you guys want a spoon?
Yeah, cut no, not one cut in the middle, and then you cut just like but like.
You sure you don't want a spoon?
Yeah,
It doesn't hurt your lips when you eat it.
No.
Um,
I so Daddy, cut it like this.
okay, you want one too, bud?
Yeah. Can I have more than one?
Guys, you want two?
Yeah. This only has 10 calories. 10 calories.
Hey, I'm just going in and read it.
You're going into a meeting. Okay. Yeah. And then we're gonna study about cactus. And cactus have water inside them. And lots of people from cactuses even yummy. Because it's like yummy. Oh wait, ow, ow. Wait, ow.
Come on.
Okay.
I keep saying okay, but you keep being really loud.
Because we're kids.
Yeah, I understand, but just because you guys are home. Okay.
You got something. You got something.
Okay. Patrick, can you hear me?
Yes.
Well, let's go to your devices are.
I actually
More.
Yes, I could hear you and I could see you.
Device settings. All right, so the speaker is right. The microphone is right. So everything's right on there. How do you test stuff on the screen?
Patrick, can you hear me?
He's muted right now, so.
Yes, I
Yeah,
can
we
hear
both
you.
can hear you.
It seems like they're trying to talk, but
Yeah.
All right, I can't hear you, but it's not. Can you bring your camera up for a sec? Okay, can you hear me? Yes, you can hear me. I can't hear you for some reason, but okay, good. All right, thank you. We're gonna go ahead and get going then. How's
Okay.
that?
Have whoever it was that got a hold of me, if you need me to come back up for, you know,
Okay,
when
cool.
when you get further down the road.
Sounds
Okay.
good. Thanks a
All right,
lot.
yeah, you're welcome.
Okay, so why don't you guys come on over here? So can you come closer here? Maybe not too close because you want to see the screen probably. You can stay, you're settled in pretty good there. You're camping out, that's fine.
Camp, not to be confused with Cyclic AMP.
Oh my God, don't do that. Someone wrote, I just gave it an exam over at Plattsburgh. Someone wrote capital C, small AMP. If I had hair, this is why I don't have hair.
So are you teaching at Plattsburgh as well, like while you're teaching here?
Yep, I'm teaching full time there. I've been a professor there for 30 years. 30 years is a long time. So, yeah, all right. So my plan today, I don't have anything new. We have an exam coming up. It'll open tomorrow morning. That should be up in your calendar already. And it's open until Sunday. Today I just want to review. I'll go over that practice exam we did. The answers are there, but it's good to just have a little talk about it. And then answer your questions if you have any questions. And then if you don't have any questions, we'll just go through the PowerPoint again real quick just to kind of review. And that's all I got. And we got no class on Wednesday because of the exam this week. So really next week will be the first hardcore exam in here. Hardcore lecture in here. Okay, any questions real quick before all right, I'm gonna share. See, this is what you've been seeing me stumble through while you're like, oh, come on. It's like, all right, Chrome tab, what I want isn't here. Window, I don't see what I see. I've got this open, but it's not showing up here. I'm just gonna do entire screen, see if that works. Sure. Now this, oops, too much. All right, this is not the right one. This was one of my notes, kind of a thing. Okay. First question. A bacterium is isolated from the respiratory tract of a patient that is very pathogenic. It produces a toxin that causes increased bronchial secretions that result in difficulty breathing. In researching the nature of the toxin, it's noted that increased secretions are directly related to production of IP3, increased levels of calcium in the cell. What are two possible mechanisms for the activity of this toxin? So what am I really asking here? What is the question about?
IP3 calcium like mechanisms.
Right. So what turns it on, basically? So what is a possible way to turn this on with the toxin?
Receptor tyrosine kinase.
I'm sorry?
RTK.
RTK, RTK, receptor tyrosine kinase. That's one mechanism, right? And that goes through what? Explain the whole pathway.
Okay. So that was a quiz that goes through like RTK, and RAS, RAF, MEK.
You've gone too far. Back up to the receptor again. There's a different enzyme that's going to be recruited there.
Oh, the SOS and GRP.
No, that's going down the RAS pathway. That's not what we want here. That is a pathway, yes, but that's not the one we want. We want to break down PIP2 and give us IP3. What's going to do that?
Phosphodiesterase.
No. Anyone else got an answer here? Let me bring it up.
Is it the PKC?
Not the protein kinase C. You're getting
PLC.
it. PLC, right.
Okay.
So you could do that. Where is it? Through this mechanism. Okay. So your answer then was this is the way you want to answer it, right? It would be a drug or a hormone or a toxin. It's going to activate the receptor tyrosine kinase and recruit up protein kinase C gamma. Make sure you add the gamma because later we talk about a beta, and that will break this down. That's one answer. Any other? What else? What's another way we can break down PIP2 and get IP3? I think we talked about 3 different mechanisms already. So I love questions like this because there are a lot of different answers. I just want to see your thought process. So as we were going through this was the first time we talked about it. And then we get down to Wnt, and there's the canonical Wnt pathway. But then Wnt also has got a G protein pathway that goes through phospholipase C. Okay, and then much later, we talk about this G protein mechanism. You've got another receptor, it goes to GQ, shouldn't be P, should be Q, activating phospholipase C, doing the same thing. Okay, so you could have a hormone activating this, the Wnt receptor, or the RTK. In addition, you could have a toxin that would directly activate this. I didn't say this in class, I should have, but if you remember, the GS and GI, right? Let's back up here. This guy here, this was alpha GS, right? If you look down at the notes, alpha GS, right? So there's a toxin, cholera toxin, right? Cholera. Cholera toxin will permanently activate this GS. So that's another mechanism that toxins can use. A third mechanism that toxins, another thing with cyclic AMP, and that will cause stimulation of cyclic AMP, right? That's how cholera causes you to, have diarrhea and then end up dying that way, because it totally turns on cyclic AMP in your intestines. You don't absorb water and you have diarrhea. Pertussis toxin does the same thing through GI. Pertussis is a respiratory thing, but it's a toxin that will permanently activate GI, and then again you get permanently inactivate GI, and then again you're getting cyclic AMP. Okay, so that's another general mechanism that the toxin from this could be doing. It could be permanently activating GQ, right? Or it could directly activate phospholipase C. So again, the kind of brainstorm activity I want is a drug, a toxin, a mutation could affect any protein in the pathway. Just explain to me how that mutation or that toxin activated this protein, that turns on everything downstream, inhibited this protein, turns off everything downstream.
How does cystic fibrosis play into this?
It doesn't play into this one. Cystic fibrosis is, it's a mutated protein that's a chloride channel. And normally when sodium is secreted, chloride will go with it, making water and mucus. And then that's coming out of your lungs, cleaning your lungs constantly, right? If you don't have that chloride, then the water doesn't go with it. The mucus gets really thick and it gets stuck in your lungs. And that's a lot more infections because you're not cleaning the lungs, and it goes in like that. So how does this relate to the lungs? Basically, well, I don't know. I lost my track. I lost my train here. Okay, are we good with this? Any questions about this? Again, almost all these questions are kind of hypothesis generation questions. There's several different answers, and I just want to see how you're thinking about it. All right, this is a sneaky one. I gave this to my undergrads. I don't think anybody got it right. Type 2 diabetes is a disease that causes a person to become less responsive to insulin. A gene that is mutated in familial type 2 diabetes is TCF7L2. This is a transcription factor that binds to beta-catenin in the nucleus to repress the gene for pro-glucagon, which is going to be made into glucagon. Why might this produce symptoms of diabetes? Okay, so let's go to, so at this point, you should know the mechanism of the insulin receptor, although this is something we didn't talk about. So it's adding something in addition to what you should know already. There's my insulin receptor. Okay, if you remember this insulin receptor, sure, go ahead.
Is this being recorded?
It should be, no? Let me go back to Teams, not Teams. Let me see what the heck I'm doing.
I think so.
Oh, stop sharing. Am I recording?
It looks like it is. There's a red dot in there, so.
It's not right. Oh, there. Yeah. Oh, I see. Red dot. Yeah. So it is recording.
Okay.
It looks like it's recording on my end. Thank you, David. Okay, so we're good. What the hell was I talking about?
Insulin.
Here we are. Okay. So insulin mechanism. So this question relies on you knowing the insulin mechanism, but it's going to take you in a different direction. So what do you think? What is the connection between insulin and what we're talking about here.
This is just a guess, but I think that mechanism activates the GLUT transporters.
Mhm.
So if they're not getting activated, then the glucose isn't being absorbed and converted into glucagon. So there's just going to be a lot
No,
of.
glucose isn't converting to glucagon. Glucagon's a protein,
Okay.
so it's going to be activated by, here's a heavy hit, it's going to be activated by beta-catenin binding to this transcription factor, and that's going to turn on production of glucagon. What's this over here? This is your connection to insulin here. What do you know about beta-catenin? So if we go back to our insulin receptor mechanism, central to it is GSK3 beta. Remember GSK3 beta? It's GSK3 beta is inhibited by AKT. And in this mechanism, we talked about AKT inhibiting this, which allows glycogen synthase to then go ahead and make glycogen, okay? But GSK3 beta also phosphorylates beta-catenin. So that's your tie-in between the insulin receptor and glucagon, okay? When you got insulin there, excuse me, you are doing what we talked about with the insulin mechanism, putting the GLUT transporter up, bringing glucose in, activating glycogen synthase, making glycogen, but you're also allowing beta-catenin to increase, and that's going to go into the nucleus and inhibit the production of glucagon. Glucagon, this question assumes you know what glucagon is. Maybe I should make that more clear. But glucagon is like the opposite of insulin. It's going to go to the liver, cause the breakdown of glycogen, production of glucose. So you can imagine when you have insulin, glucagon would be the opposite, so you want to repress it. So that's what's going on here. Glucagon is being repressed by the insulin being there. But in this familial type 2 diabetes, you don't have that repression. So glucagon is still being made, and you're getting, higher glucose than you should when you've got insulin down. Makes you seem insulin insensitive, right? You've heard of insulin insensitive? That's your type 2 diabetes. That makes sense? Yep.
So just like to overview, so the mutation in the familial type 2 diabetes is a mutation of the beta-catenin. So if the beta-catenin is, okay, so if that's mutated, then proglucagon is not being transcribed, so we're not creating glucagon, so we're gonna have higher blood sugar.
Right, exactly. There's only one thing that you are wrong, and I didn't want to interrupt you. This isn't mutated. This is a co-repressor with beta-catenin. So if this is absent and this builds up,
Okay.
you're not going to repress anymore. Boy, this is harder than I thought. I thought you guys would be. All right, shall we move on? We shall. Oh, I'm a pharmacologist, right? As a pharmacologist, you were researching the effect of the new drug DS, that's my initials, DS4532, and that's my lucky number, so you might want to save that. It's probably my password somewhere. The cyclic AMP levels in isolated ventricular cells. DS4532 alone increases cyclic AMP levels slightly from baseline. However, its main effect is to magnify the effect of cyclic AMP elevation produced by epinephrine above what epinephrine alone produces, and prolong the cyclic AMP elevation after epinephrine is removed. So you can see here, this is in a cell, we're measuring cyclic AMP, and this is time. Epinephrine gives us this response. We wash it out. This drug gives us a little bump, but when we add epinephrine, it's a lot higher than it was with epinephrine alone. And when we wash out, but we keep that drug there, it takes a lot longer for it to wash out. And here you can see the peaks here. Propose a mechanism. So what am I asking about here? What is the question?
I'm assuming it's going to be something to do with beta receptors.
could be beta receptors. A lot of receptors that link to cyclic AMP, but ventricular cells, I would say probably beta receptors. Yeah. What else? What's kind of the crux of this question? What am I really asking? Basically, what I'm asking is, with this mechanism, how could you produce the results we're seeing here through this cyclic AMP beta receptor mechanism?
Could you maybe be inhibiting the phosphodiesterase that breaks down cyclic AMP?
That's the easiest answer. That's definitely right. Yep. That's the easiest answer in terms of explanation. You're blocking PDE, and so the cyclic AMP, the baseline level will come up a little bit, but the stimulated level will come up a lot. What else could we do here? Remember what I said about the pertussis toxin stuff. So now we're getting into kind of crazy drug mechanisms that could all be true. I mean, it could be binding directly to the receptor, right? Could just be a beta agonist. No, it couldn't, because if it was, it would go up as high as epinephrine, right? So it couldn't be an agonist at the receptor. It could be inhibiting the GTPase activity of the alpha subunit, right? Because then the alpha subunit would stay on longer. So baseline is going to come up a little bit, stimulate is going to come up a lot. It could be increasing the affinity of adenyl cyclase for the alpha receptor or the alpha subunit. That again is not going to allow it to be released, and so it's going to stay on longer. Or it could be allosterically modulating the receptor, right, causing it to have a higher affinity or link better to the G protein, something like that. So again, the basic idea here is when you read the question, you're like, oh, he's talking about cyclic AMP. Just like, are you Matt?
Yeah.
Matt. Just like Matt said, oh, it must be beta receptors. Well, first thing you want to do is in your mind go, what is that mechanism? How does it work, right? That's the memorization aspect, right? You know that thing. Okay, now how can we change this, give us the result we're looking at? Easiest way is phosphodiesterase. We're just going to block that. It's going to do this. But any protein in the mechanism could be a receptor for that drug. You just have to rationalize how it could actually produce that effect. So say you were doing this experiment, you found out it wasn't PDE. What else could it be? So it's hypothesis generation. And this is like very similar question. Receptor tyrosine kinase pathways often lead to cell division. Cancers can often begin when a mutation in the gene results in the loss of control of some proteins in the RTK pathway. Choose a protein in this pathway that could be mutated to cause cancer. Describe whether the change in its activity results in constant activation or constant inhibition, leading to constant activation of the pathway. So again, hypothesis generation. Look at all the proteins in the RTK pathway, and we've got 3 different RTK pathways. Pick any one of those and say, well, this one has been mutated, and now it's out of control. Anyone want to take a stab at this one? Again, when you're taking the test, first thing you want to do is just recall the pathway. Put this in your head. Oh, go ahead.
That's not what I was thinking.
Okay.
Because if we're talking about cancer, though, couldn't you also suggest that it's like with the AKT leading to P53?
It could also be that pathway. Yep. could be either pathway. Or it could be Wnt. No, we said RTK though. We said RTK. So it couldn't be a Wnt pathway. So again, like any protein here, most obvious is the hormone, right? The cell could be mutated, so it's producing hormone and releasing it. You're getting autocoid stimulation. It could be overproduction of the receptor. Wouldn't know this probably until next chapter when we talk about it. But if you've got an overproduction of the receptor, they activate themselves, probably just by bumping into each other without hormone. PI3 kinase, I don't know how it could get activated without a receptor. I don't know, but AKT could be, mutated so it's constitutively active. Axin could be mutated out so it doesn't exist anymore. That's gonna inhibit GSK3 beta, let beta catenin build up. APC, same thing. If it's mutated out so it doesn't exist, beta catenin builds up. We go back to this mechanism, again, the hormone or the receptor. Here's a common mutated gene in cancers, RAS. So there's a mutation in RAS where it no longer has GTPase activity. It can't break down G2P, so it's constantly activated. MAP kinase is another common one. It's constitutively activated by a mutation. So it can never shut off, so it's always on. But you could say the same thing for MEK or RAF. So basically, the bottom line to this question is anything that is positively activating the cell division, if it's mutated to be constitutively active, then it's no longer connected to hormone and the cell just keeps dividing. If it's inhibiting the division somehow, like this thing, right? This is inhibiting cell division by breaking this down. If any of these are missing, then this builds up and the cell is active.
I just have because I don't think the people on the Zoom can see you
Oh.
touching the screen. So.
Sorry, you're right. I get so excited. So anyway, what I was saying was, if any of these things are missing, then beta catenin is going to build up. So these are kind of tumor suppressors, basically. We'll talk about that in the next chapter. But anything in this pathway that gets constitutively active will then cause the cell to divide. So if it's a positive growth regulator and it gets constitutively active, then the cell is going to keep dividing. If it's a negative growth regulator, something that shuts things off, it'll get stimulated that way too if it's missing. So we talked too back here about some of the things that get activated by these SH2 domains. One of them is GAP, right? GAP is the protein, GTPase activating protein that causes RAS to break down GTP. If this is mutated out, right, both copies, you lose both copies of that, the cell becomes active, right? Because you can't shut RAS off. We also talked about, in terms of tumor suppressors, PTP here, phosphotyrosine phosphatase, that is going to take the phosphates off RTKs. So that's a negative cell regulator. That if that's gone, then the cell's going to keep dividing. Another tumor suppressor. This guy, P10, right? P10 also has an SH2 domain, and when you activate RTKs, it comes up to the membrane, and it's the negative feedback. It's going to take that phosphate off PIP3, turn it back into PIP2. So if this is mutated out, the cell keeps dividing. Okay, again, the mental framework here is, what is the mechanism I'm looking at? Positive growth regulators could be mutated to be constitutively active. Negative growth regulators could be deleted, and then they're no longer inhibiting. And that'll probably make a lot more sense when we get into the next chapter and we actually start talking about it.
If this question was asked on the test, would you want us to talk about all of those pathways or just stick to one?
I'll ask for one or two.
Okay.
I won't ask for all of them. What did I ask for here? Choose a protein. So here I'm only asking for one. By the way, make sure you read the question. That way you're going to be up on top of my undergrads and just answer whatever they want. Whatever the question is, it doesn't matter. I studied this. I learned it. I'm going to tell it to you. Okay, thank you. Thank you. Okay. That's all I got for warm up here. You want me to go through all these notes again, just for the heck of it? Okay, and I'm just going to like emphasize the highlights here. All right, the first part is really general. Ligand, receptor, signal transduction, changes in gene expression. Small molecules, proteins, and hydrophobic molecules. Cell surface versus intracellular, this is all just general stuff. There was a question about this, a multiple choice question. This is pretty easy. Endocrine, paracrine, and autocrine, right? And juxtacrine. Those are your different ways that hormones work. All right, now this is kind of interesting. This is like, why is it like when you have something like epinephrine, right? It's a hormone systemically released. Why is it some cells respond in one way, other cells respond in other ways? Excuse me, it's because you've got a lot of different subtypes of receptors. Some cells are not going to have any receptors for epinephrine. Some will have different types of receptors. And those different types of receptors can be linked to different responses in the cell. So it's all kind of modular. You can do anything you want with these receptor mechanisms anyway. This is specifically for your large heterotrimeric G protein mechanisms. They're very flexible, really, from cell to cell. Okay, steroids, cholesterol, retinoic acid, right? Vitamin A gets converted to retinol. First retinol to retinal to retinoic acid, so it's being dehydrogenated. And then it's usually autocoid, activating itself, cell activating itself, but it can also be released into the neighborhood, and other cells can respond to it. Use thyroid hormone. Yes.
I'm forgetting the exact question on the quiz, but I think there was a question about hormones and which ones maybe were hydrophilic or hydrophobic, something along those lines. And when I was going through the notes, it seemed like all of them were. One of them was a precursor, I think, and I don't know if that's why that was the correct answer, but I'm just forgetting the exact question.
Never assume that I wrote a good question. That's not always the case. Let's bring that up real quick.
I think it was on the first one.
Oh, on the first test?
Yeah. Or not the first test, but the first quiz for this section. Yeah, it was that first one. Or not it was like lipophilic. Yeah, 6.
Which is not lipophilic. Thyroglobin is not lipophilic. It gets digested. It's a sneaky one. It's digested into thyroxine.
So, because thyroxine is a lipophilic hormone, right? And is it because it's thyroglobin is a precursor?
Yeah, it's a precursor. It's not the hormone.
Okay. Yeah. I was just confused because I think, isn't retinoic acid a precursor as well?
No, that is the hormone. Let's go to that. So retinol gets converted to retinol.
Oh, okay.
And that's the hormone. But thyroglobin is this big protein over here, which then gets broken down into thyroxine. This is a sneaky question, I'll admit. But you guys did so well in the first exam, I think I pushed you a little more.
Thank you.
Sure. All right, so thyroid hormones. There was a multiple choice question about this too, which seemed a little picky. I don't know, but I had to come up with another question. There aren't that many things to ask about this chapter. So anyway, it was like glucocorticoids, like cortisol, will bind to the receptor in the cytoplasm, and you've got these heat shock proteins and IP that are kind of holding it until it does. Then it goes in the nucleus. Here, the receptor is already in the nucleus, heat shock protein holding it, and then you've got this hat coming in here, histone acetyltransferase, and that puts acetate groups on the DNA and causes the histones to let go. Oh no, that puts acetates on the histones and they let go, opens up the DNA so you can get transcription. And here's the other side of that, deacetylase. So this is turning genes off, this is turning genes on. So it's a co-repressor here in the thyroid hormones. And here's a co-activator in the thyroid hormones. Here it's just a co-activator. Histamine receptors. My favorite ones are the H2s. I'm enjoying this class today because I'm on omeprazole. I'd be miserable without it. H1 is really important in inflammation. We'll talk about that in a second. H2 is more the stomach. H3 are central, not that important. H4 are really important in the inflammation response too, which is this. You know, first your B cells get exposed to some antigen, they produce IgE, and then your mast cells have got receptors for the constant part of the IgE, the Fc region. So the Fc region then binds to your mast cells. When you're re-exposed to that allergen, that clumps the receptors together and activates the release of IP3 calcium, which is like friggin' everywhere, right? That mechanism is all over the place. So IP3 calcium then causes these granules, which are filled with histamine and also a lot of proteases, release, and then that starts the inflammation process there. So that's kind of important. Here's our eicosanoids. I don't expect you to memorize all this stuff. What would be nice to know though is arachidonic acid, typically the second lipid in a phospholipid, and the main pathways it's going to take is cyclooxygenase, which is going to make prostaglandins and thromboxanes. Prostaglandins are really important local mediators. They're going to cause inflammation as well. They can cause pain, a lot of that local inflammation response. Leukotrienes, on the other hand, are lipoxygenase, and that's going to activate cells of the immune system. And then cytochrome P450 epoxides, honestly, it's the first I'm hearing about it, so let's pretend it isn't there. We could black it out, but whatever. And that's just the same pathways. Again, don't memorize this. Just know the general idea that arachidonic acid, lipoxygenase this way, COX this way. Electrical synapses, probably the simplest thing in the world. You just have channels between two cells that allow things to go through. Small molecules can go through. Mostly I think of these in terms of ions going through and passing electrical current from one side to the other. But you could also have, you know, ATP going through or other small molecules. But mainly we're talking about it in terms of channels or electrical synapses. And then you have the word syncytium, which I love because of all the Y's in it, syncytium. So you should know that word. Syzygy. Anyone know what syzygy is? It's a perfect alignment of three stars. SYZYGY. It's a great word. Question?
This might be kind of a stupid question, but when you're talking about electrical synapses, I assume you're talking about like neurons or cardiac cells, or is there something specific here?
Nope. I always think of it in terms of cardiac cells.
Okay.
Also, connection between smooth muscle and endothelial cells. mainly because that's what I know, that's my background. They also exist in the CNS, probably a lot of other cells I don't know about. But I mostly think about it in terms of the heart, the smooth muscle, and the CNS. Yeah. Chemical synapses. So your presynaptic bulb here, you've got these vesicles that have transporters to put neurotransmitter in. They also have enzymes to make neurotransmitter if you're pumping precursors in. This always reminds me of catecholamines because tyrosine will get pumped in, it'll get converted into L-DOPA, L-DOPA gets converted into dopamine, dopamine gets converted into norepinephrine. So that's kind of what's in my head when I'm looking at this. You've got an enzyme inside that's going to do conversion of precursors into it, but this will also pump neurotransmitter that gets that's free in the cytoplasm here into the synapse, into the vesicles. Those vesicles then will dock against the presynaptic membrane, and when an action potential comes through here, they are docked. They're docked right next to these calcium channels. And when the action potential comes down, calcium comes in, and that causes all these vesicles to move forward, and the front ones to fuse, releasing neurotransmitter, activating the postsynaptic receptors. And always remember our presynaptic autoreceptors. And this is making sure, if the synapse is full of neurotransmitter, we don't need to be pumping more neurotransmitter out. So when this is full, we shut off or slow down at least release of neurotransmitter. This mechanism, by the way, I don't have it drawn in here. Never mind. It's not going to be important in this class, but basically this mechanism is this. That presynaptic autoreceptor is activating GI and inhibiting the production of cyclic AMP. That's not that important here. That's not what we're talking about. But anyway, just so you know, we're doing this by decreasing cyclic AMP. Should I not throw those little things out? Sometimes I'm afraid I might be confusing you with adding extras. I don't know. Anyway. Okay, and then here's vesicle fusing. This is from your book. I'm not crazy about this drawing. I like this drawing better, which is from a different book. Basically, your vesicle is going to have a V snare on it. Snares are the mechanism to fuse. And Rab GTPase. So Rab is another small monomeric G protein like RAC-ROS, CDC42, RAS. So Rab GTPase is there on the vesicle as well. The vesicle comes down and docks, like we said above. Right? Here is our docking to the membrane. So those are the docking proteins. So it docks to the membrane, and then it kind of primes where your snares are attached, but not really fused. And then when calcium comes in, Rab GTPase hydrolyzes GTP and causes them to fuse, and then that causes the membranes to fuse, and you get exocytosis here, release of neurotransmitter. And then to recycle, you've got NSF and the SNAPs that are going to kind of unfuse the V snare and the T snare and allow it to release. And that's because you're going to have to bring all this stuff back in and then reuse it. So those T snares the target snare, which is on this membrane, will stay in this membrane. The V snares will get recycled back to these vesicles. So they'll be on the vesicles and ready to release again.
Is this the one we just went over? Is that the mechanism for exocytosis everywhere or just in the synapse?
That's everywhere. That's everywhere. Yeah. And just adding a little more on top of this, botulinum toxin, right? Botulinum toxin will cut these snares, right? It'll digest the snares, and then vesicles can't fuse and release anymore. And usually you're doing that You know, when the people are doing it cosmetically, they're doing it in the face to paralyze those muscles. It'll paralyze that neuromuscular junction. And then those muscles don't contract and you don't get that wrinkles in your forehead. It does nothing for a unibrow though. I've tried, believe me. Joke, I'm sorry. Okay. So there's your snares. This figure's there just because I think it's pretty. Okay, once you release neurotransmitter, how does that stop, right? You release it and you want to get rid of it. So you're doing pulses of neurotransmitter, not just filling the synapse constantly. So number one, the most common mechanism is reuptake. And this is a sodium amine cotransporter. We call it a pump, amine pump, but it's not an ATPase, it's a sodium cotransporter. And you've got the same kind of mechanism here, sodium amine cotransporter on the vesicle. This is astrocytes, which are glial cells that are helping out at the synapse. Something like glutamate will diffuse out of the synapse, be taken up by the astrocytes, which could degrade it or could just pass it back into the axon bulb to be recycled. And last but not least is our old friend acetylcholine. I think some peptides might be broken down in the synapse. I don't know. Could be wrong about that, but I know acetylcholine is. So acetylcholinesterase then breaks acetylcholine down. The choline and the acetate get transported back in. And again, we've got our enzyme there in the vesicle that is resynthesizing the neurotransmitter from the precursors. Okay, so those are the three mechanisms.
So this one is still using the same pump system, and the glial cells pushing it in. It's just degrading it on the outside instead of just recycling it directly into the cell.
Yeah. And it's also specific for the neurotransmitter too, which I believe the pumps are as well. I'm always shocked when they say there are only 20,000 genes in the human genome, because it's like every mechanism I look at has got 10 new proteins I never heard of. There's so many. But anyway, and this isn't important. This is just something I wanted to talk about.
Sorry, I didn't write it down. What did you say the name of the pump that's in purple?
It's a sodium amine cotransporter. So you're using the power of the sodium to bring it in, or the driving force of the sodium. And again, don't memorize any of this. I don't remember why I put it up there. I guess just to show the different types of neurotransmitters and what they do. But and yeah, it doesn't matter. Don't memorize that. And this I just think is cool. So this is just about how astrocytes will release gliotransporters that are going to activate neurons as well. Pretty easy. Okay. Let's do this real quick. So the first half of this chapter is all about receptor mechanisms that are going to activate gene transcription. And usually this is going to be about two things: cell division and differentiation. So differentiation is when you're turning on different genes to change the cell type. And cell division, of course, is just making more cells. And these are usually activated by GFs: epidermal growth factor, nerve growth factor, fibroblast, vascular endothelial growth factor. They usually end up in GF. So these are activating your receptor tyrosine kinases. So this is the way we think of it. This is what it actually looks like. Tyrosine's in the tail. When the hormone is around, receptors clump together, cross-phosphorylate, and that makes these SH2 binding domains. So proteins with the correct SH2 domain will bind to this receptor. And here I talk about where the name SH2 comes from. SARC, we talked about as part of the complex of the focal adhesion. SARC has got an SH3 domain. Well, this is where the idea comes from. This is the SH1 domain, which is the kinase activity. SH2 is going to bind to phosphotyrosines. SH3 binds to a polyprotein sequence. And then here's your SH2 domain. I like this because it's kind of a plug and play model where this is a domain that evolutionarily probably got swapped between different proteins and amplified as DNA does. And the difference is the amino acids here will be, the amino acids here will be specific for some of these other amino acids. You've got a phosphotyrosine plus 4 amino acids. And so your phosphotyrosine binds there, the other 4 amino acids bind here. So it's selective. And you can have multiple SH2 and SH3 domains on different proteins, and that causes recruitment. Okay, recruitment's something I love personally. Basically, it's like you activate that receptor, it brings a lot of other proteins together, recruits them to the membrane, and when they're recruited to the membrane, they come close to their substrates and they activate all their substrates, and that's kind of how the mechanism works. And this particular protein, you've got the enzyme here, sort of the GEF SOS, and then this adapter protein GRAB2. And these are always linked together by the SH3 domains in GRAB2. So this SOS then is going to activate RAS, which is a small monomeric G protein. So the GEF causes it to release GDP, it picks up GTP, and now it's active and it activates something. If it's RAS, it's activating RAF. And then this will stay on unless GAP turns it off. So here's the thing. I don't know if I stressed it enough during class. You've got 2 big classes of G proteins. You've got your small monomerics, RAC, RO, CDC42, RAS, RAB. And then you've got your large heterotrimerics, right? Which is alpha GS, alpha GI, alpha GQ. And the big difference between these two for me, one big difference is the large heterotrimerics are heterotrimeric. You got that beta gamma. Sometimes that beta gamma has activity too. But also the large heterotrimeric, the alpha subunit from that will break down GTP pretty rapidly. Okay, so it doesn't need a gap. It's fine without a gap. This needs a gap. So this is going to be a tumor suppressor, GTP activating protein, because you need that there to shut off RAS. That's a big difference between those two. Okay, but anyway, here's your recruitment. RAS is a protein that has got a lipid attachment, so it's always at the membrane. So when this gets activated, it brings GRAB2 and SOS up to the membrane, and then it interacts with its substrate RAS and turns it on. And then here's that whole pathway. I asked a question about this on the last quiz, didn't I? Order these. So there's that. And in the next chapter, we'll start talking about this. Once you activate these transcription factors, how you can turn the cell on to start dividing. So cell division is next. And then spring break, right? Kind of sucks. UVM and Plattsburgh don't overlap, so I don't get a spring break this year. It's horrible. I'm gonna have to work like the whole semester. I haven't done that in 30 years. Oh, never listen to a professor complain. Their lives are too easy. Okay, so this just shows some of the negative feedback pathways. Let's just say we don't need to worry about this too much. Wink, wink. Okay, so here's your other pathway, the PIP3 PI kinase. PI3 kinase, PIP3. So basically, PI kinase is going to phosphorylate PIP2 into PIP3. And then anything with the Plex string homology domain is going to come up to the membrane. Again, we're recruiting here. What did I want to say about? So again, you know, if you look at this, all these phosphates, that's a big negative thing sticking right there, and it's attracting this to come up and bind to it. And again, like with all these recruitment mechanisms, now they're near each other, right? So PDK1, AKT is its substrate, so they're next to each other. mTOR, it's going to phosphorylate it. This gets double phosphorylated, and then it becomes active.
So without, if you had PDK1 but no mTOR, you still wouldn't have activation of AKT.
I think that's true. I'm not 100% on that, but I think that's true. I think it needs to be phosphorylated by both.
And then just in the last slide, the green and gray with the phosphate on it, is that just a different depiction of an RTK? Right?
Where we
Yeah,
talking?
that whole,
This?
no, like the one that's in the membrane, it looks like. Is that just another?
No, this is the phospholipid. So this is your glycerol, 1, 2, 3 carbon, and 2 fatty acid chains. This is an inositol sticking out. Okay.
So where is like the RTK in the
Right.
pH domain?
It's not. What's going to happen is your PI3 kinase is going to make that, and then other things with pH domains come up. Okay. I know it's confusing because every time they draw it, everyone draws it differently. Let me show you something real quick. Maybe this will make it more clear. Maybe not, but. All the way back in chapter 2, we talked about this. So this was one of the first things we talked about in chapter 2. Here's your phospholipid, right? This is pretty ugly too, but you've got a, okay, here's your glycerol, 2 fatty acid chains, and inositol here. Phosphatidylinositol there. Okay. So fatty acid chains, glycerol, inositol. Okay, so anyway, that activates this mechanism, and this mechanism is going to block GSK3 beta. GSK3 beta and beta catenin are central to so many different things. It's an important one to know. Do you guys take developmental biology as part of this? No? Okay, it's important there too. Okay, so GSK3 beta, when AKT gets activated, we're blocking it, and that allows beta catenin to build up. Because normally GSK3 beta is phosphorylating beta catenin, and that causes it to be ubiquitinated and destroyed. We'll see that again in the Wnt mechanism. At this point, it's the same as Wnt mechanism, but upstream it's different. What's next? Oh, this is important too. Okay, so this is mainly what we think about in terms of the PI3 kinase mechanism. The receptor tyrosine kinase clumps together, autophosphorylates or transphosphorylates. It recruits PI3 kinase up. We make PIP3. That activates these kinases, which then inhibit GSK3 beta. And then you get increase in beta catenin and cell division. But as I said before, when you activate cell division, you also have to block apoptosis. Because if the cell's hyper stimulated, it will typically go into apoptosis, kind of as a failsafe. So here you can see with AKT, here's the GSK3 beta, which is allowing beta catenin, which is bringing up c-Myc and cyclin D. We'll talk about them in the next chapter, but they're going to cause cell cycle. Also, it's going to block these guys that we talk about in the next chapter, which is CDK inhibitors, P27 and P21. And here's a really important one. It's going to inhibit FOXO. FOXO is going to block cell division here this way, but also increase apoptosis by upregulating Fas ligand and BIM. And again, it's hard to teach cell biology because everything's so interconnected. We haven't talked about apoptosis yet. It's 2 chapters from now. But just take it on faith that I haven't lied to you yet. And Fas ligand and BIM and BAD are all associated with apoptosis. So AKT is blocking apoptosis through FOXO and by directly inhibiting BAD, and also by activating MDM2. Remember, when you've got an arrow, it's activating. When you've got a block, it's blocking. So it activates MDM2, which blocks P53, and P53 causes apoptosis. So 3 different mechanisms we're using here to block apoptosis. And then through TSC1, 2, we're increasing protein synthesis. We've got another mTOR here just to make life complicated. Life is so complicated, isn't it? Get inside a cell, suddenly it's like you're talking about rocket science. Cell biology is so much more complicated. Look, we got to the moon in 9 years, right? We still haven't cured cancer. You know, cell biology is so much more complicated. Okay. Oh, and here's the insulin mechanism. I have an animation for this. Doesn't work. I should remake it because it makes so much more sense when it's moving. But anyway, your receptor's here. Hormone binds to it. The big difference between this and the other PI3 kinase mechanism is you've got an intermediate protein here, insulin receptor substrate 1. That comes up and gets phosphorylated, and then that brings PI3 kinase up. You get PIP3 made again, and this part's all the same, PDK1, PDK2. The only difference here is with the insulin mechanism, AKT is going to cause fusion of this vesicle by phosphorylating AS160. And it's also going to inhibit GSK3 beta, glycogen synthase kinase 3 beta. So this is glycogen synthase. So this is where you learn what that name actually comes from. Glycogen synthase kinase. It's phosphorylated glycogen synthase. So normally this is inhibiting glycogen synthase. When AKT is active, it's going to block this, and we're going to make glycogen. And also, you're probably getting beta-catenin activated too.
So I guess it seems I probably should have already known this. So but if something in general in one of these mechanisms phosphorylates something else, it's usually an inhibition of what it's phosphorylating.
It could go either way.
Okay.
That's why you have to know the mechanism, because phosphorylation can turn something on or turn something off. It's basically just causing a conformational change. Okay. And I think we talked about this earlier. Just another mechanism activated by activated RTKs. PI3 kinase. This is wrong. PLC gamma. Sorry about that. Come on, baby. Why did they put the most important one way at the bottom? There we are. Okay. Well, this is a protein. This shouldn't say protein kinase C. This should say PLC.
That's why I was confused that last time.
Yeah, this is wrong. Give me a second, I can fix this. Just that. You're getting a look behind the curtain. Come on, man. More or less. Yeah, that was a bad typo. This is phospholipase C gamma. I don't know. I looked at that so many times. Here it is right here. This is correct. That changes one. I don't know how they got to be protein kinase C. Here's PKC over here, activated by diacylglycerol. We haven't talked much about PKC. It can also activate cell division, but we'll get to that probably in the next chapter. And then this is just a summary of the 3 different pathways we talked about that are activated by receptor tyrosine kinases. By the way, if you ever need a PowerPoint made, I do it on the side for $30 an hour. Here's your JAK state pathway. 2 things that are interesting about this. One is that the kinase is not part of the receptor. It's a separate protein. The other thing is, I love this, the way the stats have got both an SH2 binding domain and an SH2 domain. The SH2 domain brings it up to the activated receptor. It gets phosphorylated on an SH2 binding domain, and then it dimerizes and becomes a transcription factor. Pretty simple, pretty straightforward. Kind of like that mechanism.
The first part that you just said about them being 2 different receptors, did you mean instead of it like the 2 tyrosine RTKs phosphorylating each other, it's 2 different coming together?
No, what I mean is like the receptor doesn't have a kinase, but it attracts TK2 and JAK1. And I think there are 3 different JAKs, GA1 through GA3, GAK1 through GAK3. So it's attracting those. Now, I don't know what the difference is between JAK1, 2, and 3. Probably different cells. I don't know that much. But the thing is, it's a separate protein. Okay. Anybody know what a SMAD is? What's the matter with you people? Oh, that's bad. All right. So anyway, serine threonine kinase receptors, again, really important in embryo development. You've got 2 different receptors here, the type 1 and the type 2. Type 1 will bind to the hormone, and the type 2 then binds to the type 1 transphosphorylation, and then it's going to attract the receptor SMAD to be phosphorylated. And then SMAD4 will bind to that receptor SMAD. One thing that's a little bit funky about this is that you've got 2 different kind of groups of SMADs. SMAD 1, 5, and 8 activate one set of genes, and SMADs 2 and 3 activate a different set of genes. So you can have differences between different TGF beta-like signaling molecules.
And whatever SMAD is recruited is dependent on what the type 1 receptor, or the type 1 receptor is bound to.
Right. It depends on which type 1 receptor it is. Like the type 1 receptor for TGF beta will activate these SMADs.
Yeah.
The type 1 receptor for BMP4 will activate the other ones.
What is SMAD4 activated by? I don't see it in either of those groups.
I'm sorry, can you say it again?
So SMAD 1, 5, and 8 are activated by BMP4, and then 2 and 3 are by TGF beta, but what about SMAD4?
SMAD4 is just, it's the co-SMAD to all of these. So that'll join with either of them. All right, I think we're almost done here. We're out of time. I like to throw this in just because it's medical, right? When Wnt signaling is messed up in the colon epithelium, you get epithelial cancer in your colon. And again, we've got our destruction complex, which basically the same as the AKT mechanism, except we've added some more components here that are part of it. Casein kinase, GSK3 beta, they're both phosphorylate beta catenin. This guy here, beta TRCP, is going to add ubiquitin. Ubiquitin ligase. Here's your proteasome. I don't think any of this really needs to be here. Just kind of complicates it. But anyway, what matters? I'm sorry. I get off on the aesthetics of the figure sometimes. What matters is the receptor for Wnt is frizzled. When frizzled binds to Wnt, the co-receptor LRP comes in, and that breaks up the destruction complex. So now GSK3 beta is no longer phosphorylating beta catenin, and it builds up in itself, just like before. And we've got our two other Wnt pathways. This one is just phospholipase C again. I think this is phospholipase C gamma beta, which is activated by the large G protein GQ. Disylglycerol, blah, blah, blah. This is the planar cell polarity pathway, which is involved in chemotaxis, right? So it's directed movement in one direction. And what I like about this is it kind of reinforces what we talked about in chapter 3 with RAC and Rho being activated and causing remodeling of the actin cytoskeleton, stress fibers, lamellipodia. The cell starts moving in that direction.
The LRP and frizzled, those are not RTKs, correct?
No.
Just double check.
Right. Hedgehog. Basically, the center of Hedgehog is this guy, Glee. I should get rid of that CI. CI is the same protein in Drosophila, but we don't care about Drosophila. They don't come into the clinic. So anyway, the neat thing about this protein is it's a transcription factor, but it gets bound up and blocked by these proteins, fusion, suppressor fusion, and COS2. COS2 is a kinesin-like protein, so it attaches this whole complex to microtubules. And those microtubules are in a primary cilium, not shown in this figure, but they're sticking out of the cell a little bit on a cilium. And it'll stay like that until smoothen becomes active. Patched is blocking smoothened. And because it's in this complex, it's going to get phosphorylated by these kinases and then cleaved. And this half of the transcription factor attaches to DNA. This half activates transcription. So when it cleaves in half, you only get the DNA binding part of it. And so it's blocking access to anything else. So it's blocking gene activation. When Hedgehog is around, it binds to patched, and that stops blocking smoothened. Smoothened then breaks up this complex, and Glee gets released intact. And now it's a transcriptional activator instead of repressor.
So HH and SHH are like canonical. I mean, interchangeable is what I was saying.
I'm sorry, what?
Like HH and SHH are interchangeable.
Right. If you look down here, Hedgehog has got 3 variants: Desert, Indian, and Sonic. So SHH is Sonic Hedgehog. And that's the main one. That's the most important one. The other ones, I looked it up on Wikipedia. The other ones are involved in fewer processes than Sonic. Sonic is a lot more central. And here's just one of the things Sonic Hedgehog does. Sometimes your book bothers me because it's like it gives this big figure about this, and this is just one of the many things, and there's more important things, but okay, that's what you like. So here, endothelial to mesenchymal transition is kind of interesting. It's mostly involved in embryo development. You know, epithelial cells are sheets, and the cells are grabbing each other with cadherins. And then when they get acted on by certain hormones, they let go of each other and start to crawl. And so that mesenchymal cells are released from the tissue and crawling around. Endothelial cells are bound to each other. So a notch can cause these epithelial cells to let go and start to crawl. And in this example, they're going into this cardiac jelly and making valves, I guess. But the point is your EMT transition that can occur from the loss of cadherins. Here, this is a really simple pathway. Again, it's juxtacrine. The ligand, more or less, is attached to one cell. It could be delta or jagged. And then the receptor is notch. When the ligand hits the receptor, you get a cleavage to take that off, and then another cleavage to take this off. And then this is the N-terminal intracellular domain, which is a transcription factor. So it's pretty simple. just juxtacrine signaling, release of the the transcription activator here. What's neat about this, it turns on other transcription factors like snail, and that causes a loss of cadherins, and that causes the cells to let go of each other. And that's just one example. All right, we're over time a bit. Do you want me to go through all these G protein coupled receptors? We kind of talked about them a lot already. Do you mind if I? You're okay back there? We'll just buzz through this because I did talk about them a lot today already. Bum, 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 bum. Again, don't memorize this. This is just examples. Here's this is really important. This just keeps coming up and coming up. And there's your IP3 calcium mechanism again. Calmodulin, which is the island next to coagulin. Just kidding. Okay, more stuff, more stuff. Oh, and then we've got production of NO. This is kind of interesting. IP3 calcium again. This time calmodulin activates NO synthase. NO synthase then makes nitric oxide, which diffuses into other cells, activating guanyl cyclase, which activates protein kinase G. And I know this is really, really important in blood vessels. This is where it was discovered. It's also in the CNS, probably other places as well. Physiological antagonism. Anytime something downstream of the receptor is opposing each other, we call that physiological antagonism. This is just interesting in that it's the beta gamma subunits of activating channels. You could also have the alpha subunit activated channel, or the alpha subunit activate a kinase and the kinase activate the channel. But basically, your GPCRs can be linked to channels as well. And downregulation, this is important. So let me go through this because it's important. When the ligand binds to a GPCR, it activates an alpha subunit, which goes off and does some kind of signaling, but it also releases the beta gamma subunit. And the beta gamma is going to activate GRK, G protein coupled receptor kinase. And this is going to come up and phosphorylate the receptor. And there's a bunch, but there's some crosstalk as well. It's going to phosphorylate the active receptor, but other GPCRs that are in the area as well. And that causes the protein arrestin to come up. Arrestin is going to decrease the affinity of the ligand for the receptor, so the ligand will come off, and it's going to block the ability of G proteins to interact with this receptor. So that's desensitization. This receptor is now desensitized. If the stimulation lasts longer, this happens within minutes, okay? Within hours, you get them internalized. They'll go into these clathrin-coated pits and become internalized, and that's downregulation. And depending on the length of stimulation, the longer you stimulate, the more you downregulate, and eventually these are going to be degraded. Now, if you take that stimulation away, these downregulated receptors will sit in this, briefly, they're going to sit in this subsurface pool of vesicles, and they can be put back in, again, through that snare hypothesis, right? The same way, putting these receptors back into the membrane and then, restoring sensitivity. And this is why your heroin addicts are going to die after, this is why a lot of heroin addicts die after rehab, right? They're used to being on the street, taking this high dose of heroin or fentanyl, whatever they can get. You go into rehab, and their receptors come back. Then they go out and take that same dose, whoosh, they're way over stimulated.
Yeah, I don't want to answer my question. I was just going to ask if this is how tolerance builds up to pain meds, other type of recreational.
Exactly. This is one of the mechanisms anyway. And remember, if you've got an antagonist, this is not pharmacology. If you've got an antagonist, it's going to block activation of the receptor, which means you're going to get more receptors put into the membrane. And so you get an upregulation of receptors and supersensitivity. And people realize this with cardiac drugs because people were on beta blockers, right? And they stopped taking them suddenly, tachycardia, because those receptors got upregulated by the antagonist. And that's it. That's all there is. I think next time we're gonna do it all in one day and just give it 3 weeks off. I'm trying to go by this textbook as much as possible. Sometimes I don't agree with it. But, I mean, it's right, just the order and the whatever, I'm not crazy about that. Anyway, any questions, anything I can clarify before we say goodbye? Nothing? Let's go back. Okay, so if you're watching along at home, I'm going to re-upload these because I found a couple of typos in them. So I will be loading them up again.
I did have one more question, actually. It's not content related, but is there, when we had the first exam, there was the bio game, was that like the one and only? Or is it
Yeah.
just dependent on the exam, like how you built them?
Well, two things. Number one is my exams, my games for some reason are not working. Look at this. I don't know what's going on. It's like they're here, the things are here, you can move them. I don't know. The internet, you know? And the other thing is you guys did so well in the last exam. I don't think you need 10 bonus points. I think most of the class got over 100%. Not most, but quite a few people.
And that
More than
actually
half got A's.
was my next question, is the score that's in Brightspace, like just under the exam section, is that with the points added on?
No.
Okay.
No. Well, I can't bring it up because you see everybody else's, but
Yeah,
I'm putting those into the bonus point.
yeah. I saw there was a different category. I just wasn't sure if it was already included or if it was.
Yeah, yeah.
Sweet.
Okay, any other questions, comments?
There might be a question in the, it looks like there's something in the chat.
How do I find it?
On the,
Chat.
yeah, right.
Any questions at home? Want to send me a chat? Patrick? Okay, I guess that's it for today then. And the exam will open tomorrow morning at 8 a.m. It's open till Sunday night. So take your time. Do it right. I think it's 75 minutes, same as before. I was like gonna do 20 multiple choice questions. I thought that's much. That's a bit much. So 10 multiple choice and then 5 of the written questions. And don't waste too much time on the multiple choice. Just do them, get through them, do the written and then go back. I had one student last time who never got to the written question because he spent all the time on the multiple choice. You don't want to do that. Okay?
Thank you.
All right.